With Christmas just around the corner, what better than to explain eukaryotic DNA replication with Christmas? We may not have story time at the high school, but explaining biology with a Christmas story is the next best thing.
This is the process of DNA replication. DNA replicates in a semi-conservative manner. The experiment done by Michelson and Stahl with nitrogen isotopes in the late 1950s supported that DNA replicates semi-conservatively.
First, the enzyme helicase untwists the double helix by breaking the hydrogen bonds between complementary base pairs.
As the enzyme helicase untwists the double helix, the enzyme primase places complementary RNA nucleotides onto the template strand of DNA.
The RNA sequence laid down by primase is known as the primer. It is important because it allows DNA polymerase to bind and add nucleotides to the 3' end.
DNA polymerase adds nucleotides from the 5' to 3' direction to produce a double-stranded DNA molecule. These nucleotide sequences on the lagging strand are called Okazaki fragments and are eventually bound together by DNA ligase.
In DNA replication, the leading strand elongates continuously, but the lagging strand is synthesized discontinuously in a series of fragments previously mentioned as the Okazaki fragments.
Eukaryotic DNA replication can become pretty complicated pretty quickly. So, my goal here is to explain an analogy that is a little more cheerful. To begin, we are going to take a look at the purple strand of DNA, which serves as a template for replication. Having this strand allows primase and DNA polymerase to lay down the correct complementary nucleotides. The template DNA can be represented by the previous path that Santa and the reindeer traveled along last year. Now, Santa is able to use this previous path as a guide for this year's path. This year's path is represented by the light blue DNA strand. The previous year's path is initially very difficult to follow. To help Santa out, Rudolph leads the way and clearly marks out where Santa and the other reindeer will go. Rudolph is helicase, which unwinds the double-stranded DNA to expose each template DNA strand upstream from the replication fork. Once Rudolph has cleared the path, Santa's little helpers prepare the houses on the path, or the genes, by sprinkling them with their reindeer food. This reindeer food signals to the reindeer where to stop and drop off presents. Without the food, the reindeer would not be capable or know where to begin. The reindeer food represents the primers that are put down by primase. They serve as starting places for DNA polymerase to begin adding bases. DNA polymerase is represented by Santa and his reindeer. Using the reindeer food or primers as a base, Santa and his reindeer drop presents off along the previous year's path. The presents that Santa drops off are nucleotides that are complementary to those of the template strand. Santa drops presents off to make a new path that begins at the 5' end. Thanks for watching and have a happy holiday.